All right, I'm in a bit of a loud place right now, but I thought I would try to record a video for you, see if I can explain some things in a little bit more of a clear way. And I also have a genetics video that I might be able to show you. It's old, but it's got some good information. So when it comes to mouse genetics, the most important thing to know about is the A locus. I don't know if you know a lot about the A locus or how genetics are written, but at the basic level, you've got these things called locuses or loci. And the most important loci to know about with mouse genetics are A, B, D, C, and then P, kind of. And then you've got like all the marking genes otherwise. But on the A locus, we've got some dominants and recessives, viable yellow. This is agouti. This is non-agouti, so black. We've got like extreme black. We've got hypervariable yellow. We've got lethal yellow, which is just AY. So all of these are valuable genetics to know. And what the A locus basically does is just determines the distribution of melanistic pigments, specifically pheomelanin, which is yellow pigment, and eumelanin. You'll notice that a wild-type mouse is agouti, and agouti mice actually have individual hairs that are black on the bases and then yellow on the top. So the distribution of eumelanin and pheomelanin is on each individual hair, half and half. So we've got like this kind of rough-looking pattern because of that, where it's like kind of intermingled. It's kind of a brownish color. But then if you have an A mouse, so this is like the agouti, right? If you have just an A mouse, so like AA, then it's just solid black, except for a few fault hairs that are yellow. So you're going to have a mouse that is basically all eumelanin, all black pigment. This mouse is a mix of both of them evenly on each hair. This one is mostly black with a couple fault hairs that are yellow around the ears and the vent. And then viable yellow is brindle. And this will give you a mouse that is mostly covered in pheomelanin, yellow pigment, but they actually have agouti stripes or agouti soot. So they'll have the agouti pattern, but only in their stripes, which creates kind of a tiger look. So that's the distribution of yellow and black pigment there. And then extreme black basically is the same as normal black, where it's all black, but the difference is that all of the yellow pigment is diluted to clear or white. So it's kind of a dilution pigment. So it doesn't really change the distribution of pigment from a black pigment, but it'll make it appear darker. Then hypervariable isn't really that relevant here, but it's similar to brindle. It just has a lot of different ways of presenting itself. And then, of course, we've got lethal yellow, which is all yellow. Occasionally with a little bit of soot fault hairs, but it's all pheomelanin. So that's the most important locus to know about. And every other color that goes on top of it, all of the different dilution genes, will be a dilution of one of these. So for example, in the B locus, we have two different options for the genes that we could get, or the different alleles that we could get. These are called alleles. We have lowercase b, which is chocolate. And then we have uppercase B, which is non-chocolate or just non-agouti, non-chocolate. And you can have chocolate dilution on top of any of these. If you have, so this is how you'd write this, by the way. So let's say I have a mouse that has two dominant agouti locuses. Every single time that you write the genetic pairing on every locus, they can only have two alleles, if that makes sense. So if I write the rest of these real quick. All right. And then we've got uppercase C, we've got lowercase c h, we've got c c h, we've got lowercase c, which is just albino. We've got extreme dilution. I think those are all the most important ones, the main ones that you'll see in the fancy. And then for this one, you just have lowercase p and uppercase p. So in each of these different loci that we have, you can only have two alleles. So whenever you're doing like a genetic comparison or trying to guess what alleles you have, you'll take 2 of whatever these have, and you'll combine them together to figure out the results. So for this, I'm choosing to put 2 alleles here that are both agouti. Or if I wanted to have an agouti mouse, the reality is that agouti is dominant, hence the uppercase A. When they're represented by uppercase, that means that they're dominant, and they're the ones that are, oh, I forgot one, AT. Oh, wait, sorry, it's lowercase. There we go. And tan, sorry for the switch in focus, but a tan is basically a half and half where they've got eumelanin on top, they've got black pigment on top, and then they've got pheomelanin on bottom. So they're combination. And as you can see, it shows you where the yellow and black pigment is on the mouse. But back to this, when you're writing out your genetic codes, you can only have 2 alleles from each locus. So from the A locus, I have agouti. And let's say that from the B locus, I have 2 Bs that are lowercase, which would mean that my mouse was chocolate, because chocolate is recessive. So you need 2 Bs to get the expression. Anything that's lowercase or recessive, you need 2 copies of the gene in order to get it. If it's dominant, like agouti, you only need 1 copy. It can get a little bit more complicated with codominance. For example, tan is actually partially semi-dominant. So the tan belly, the yellow belly, will always show up. So you can have a agouti mouse with tan, and the tan belly will be there, but then the top will turn to agouti. So the black changes with whatever other A locus gene you have, but then there's always that tan belly as well. So kind of interesting. Tan is a very fascinating gene. But if I go down this list, I can pick out what 2 alleles will go with every single locus. So for the A locus, I've got 2 agouti. For this one, I've got 2 blues. And we're going to say that this mouse is not blue. So it's chocolate, it's agouti, but it is not blue. We're going to say that it also doesn't have any C dilutes, so it's uppercase C. And we're going to say that it also is uppercase P. And this symbol right here just means that you don't know what the other genetic is, or it doesn't matter. So if you see this star sign, it just means, okay, well, if it's got an uppercase P, it obviously can't be a pink eye dilute, and it doesn't really matter to what it turns into at the end, because phenotypically, we're not going to see that it has a little p. If you know about it, you can put the little p here so that you know it carries, which is more relevant if you're actually trying to figure out what you're going to get. But for like calculating stuff like this, you can just write this as a way to simplify your writing style, or if you don't know what it is and you just don't want to guess because you may or may not know. So we're going to say, I don't know if it carries pink eye dilution. All I know is it's agouti and chocolate, and then it may or may not carry those other things. But what you're going to get is a mouse that is agouti, so it's got all those different colored hairs, and on each of those individual colored hairs, the chocolate dilution is going to affect in different ways. Chocolate dilution turns black pigment, which is the bottom half of the agouti hair, into chocolate. So it'll turn it brown. So instead of a black base of the hair, it'll be a brown base of the hair. And chocolate doesn't really affect yellow very much, so the top is going to stay yellow. And that leaves you with a cinnamon mouse, which is kind of this warm, soft, ruddy sort of brown look. And it's very pretty. And then, of course, if our mouse, instead of having agouti, had non-agouti or black self, like this, but then it also had the brown or the chocolate, what we'd end up with is a mouse that was a solid brown color, hence the chocolate. Very pretty little cute mouse. I don't know how to color it in to show you brown, but imagine that mouse right there is brown. And what you can do, we have this thing called, oh dang, the name escaped me right as I was going to do it. Hopefully you know what I'm talking about. I forgot the name. Maybe I'll remember it as I'm going. Punnett square. There we go. So your Punnett square, you always just have this many tables because you're only going to do one locus at a time. So this is just me going to be, I'm just going to be figuring out the A locus and what it may or may not carry or pass on for the A locus. And what I recommend is, especially when you're starting, just do one Punnett square per locus. And what you're going to do is you're going to have a mom side and you're going to have a dad side. And you're going to put your best guesses as to what they have for each locus in here. So for the A locus, the most important locus and the one that you'll want to spend the most time guessing, you'll want to figure out, okay, do I think that they're agouti? Are they just black self? Are they extreme black? Are they brindle? Are they tan? You know, what's on their locus? And let's say that you have a mouse that looks like this. And then they're kind of striped and like a reddy brown on top. And I look at that mouse and I'm like, huh, what could that guy be? Well, he's obviously tan. So, and this is going to be our dad. And then we can put, okay, so he's obviously tan. And since he's got that reddy brown look, I'm guessing he's also a Agouti. So we know that those are our two A-locus genes. Mom, as our mouse, is just a normal black colored mouse. She's a black self. And I know that she is obviously not tan for that reason. She could be extreme black, but it's not super likely. She's obviously not brindle because brindle's dominant. She's obviously not dominant yellow because that's also dominant. So her only really good option is this right here, our normal black. So since that's our best guess, and it's not really relevant whether it's extreme or not, for most colors, we're just going to guess normal AA black self. Since it's recessive, you need 2 copies in order for it to express anyway, so it would make sense that this mouse would be AA. And then what you'll do is you will then put the 2 combos of alleles into each box to see what different combinations of alleles you could get. So for this mouse, I could get a dominant A and a lowercase a, or sorry, a Agouti or non-Agouti. This mouse right here could be a black tan. Then this mouse right here, another black tan. This mouse right here, another Agouti, because since it's got a dominant A, that'll overpower the self, and it'll make this mouse Agouti. So that tells us that we have about a 50% chance of getting a black tan and a 50% chance of getting a Agouti out of this pairing. And then, if we want to go a little bit further, we could, if we have, for example, a mouse that is chocolate. I'm not going to color it in because I don't know how to... Here, I'll just do the stripe pattern. How about that? So that's our chocolate mouse. It's just a nice soft self chocolate. And we'll say that's dad. And then let's say that the mom is a tan, but she's a black tan. So we don't actually know if she carries chocolate or not. And this is where the little stars that we drew earlier, or that I mentioned as like placeholders, are going to come in handy. Because for dad's side, mom's side, so we know for mom that she's obviously at least got tan, and she's most likely got A on this side, but she could also be tan. So we can just either put a placeholder here, or you can put a star here, since we don't actually know for sure whether or not she is full on black tan. For him, we knew that he was tan and Agouti because he had both. For her, she's a black tan, so she could be black or she could be tan again. We'll just put black self for now, since we're not positive, and it works pretty well as a placeholder. It's a likely option. And then for dad, he's chocolate. So we know that for his A locus, since he's a self, he's got AA. And that leaves us with the possibilities of AAT, AAT, AA, AA. So we know that we have a 50% chance of a tan base and a 50% chance of an A base. And then for the D locus, for these same mice, I'm going to make this chart right here. Whoops, that's pretty awkward looking. Sorry, I'm drawing through my phone. And then for dad, mom, and for this locus, it's represented by B. I know that dad is a chocolate, which means that on the B locus, he is lowercase b, lowercase b. And we know that for mom, she is not obviously chocolate, but she could carry chocolate. So we know that she at least has an uppercase b. And for this one, we're going to put a star because we don't know if she has lowercase b or not. So for this one, our possibilities are this. For this one, that's a possibility. This one, we're not sure. And this is what you would learn from a test breeding. So if you breed these two together and you get a few chocolates in the litter, you know that mom carries chocolate. So you have to test breed to know for sure what's hiding under the surface to see what these recessives are. And that's why we have stars in breeding. This is our maybe. This is what we'll find out. And once we get the actual babies in the litter, let's say that the litter from these guys looks like We've got a tan with a chocolate top. And then we have a black self. We have a black tan. And then we have a chocolate self. This tells us that this right here has to be chocolate, because the only way we'd get chocolates in the litter is if this mama carried chocolate. I don't know if that helps, but as far as Punnett squares go, this is going to be your easiest way to use it. And once again, you ever see stars? That's just a placeholder. And a lot of times when people are writing out genetics like this, that's their way of writing out. You can see here, I've got CE. That's what that is. What I actually recommend doing is this thing, because that means to the power of E, and that's more accurate for drawing this, even though it's not mathematical. But I've run out of space on this page, so I'm going to go ahead and flip, and I will real quick go into more details on the mice that you showed me. It's kind of tough because you've got some diluted recessive yellows, and recessive yellow is unique. So as I said, we've got the A locus, we've got the B locus, we've got the D locus, we've got the C locus, we've got the P locus, but we also have under that some less common loci, or loci that have less on them. We've got the E locus, which is where recessive yellow is. We've got the S locus, which is where a lot of the marking genes like banded and pied are, or at least where we speculate they are. Or is it the W locus? I think it might be W and then S next to it for spotted. So that might be the W locus. See, even I don't know everything. And Merle is on an unknown locus. We're not sure about it. We're also not sure about variegated or umbrous either. All of those are kind of speculative, but I think we just put U for umbrous locus. But yeah, so you've got a whole bunch of different loci that you can pick from to determine your color. And as I said, each locus is going to have 2 alleles on it. So your mice look to me to mostly be recessive yellows. I'm not sure where you got your mice or if they're all from the same place, but I see recessive yellow coloration throughout the majority of them. So that tells us that we've got a recessive yellow base. The cool thing about recessive yellow is if it's on top of, and this is what I speculate you've got, if it's on top of black self, it'll just turn the black self into yellow. So EE actually dilutes your A locus to yellow. Whereas normally the only way to get yellow mice is if you have like a dominant yellow, like this. But with the E locus, it's one of the few genes out there that turns black to yellow. So it's a very fascinating genetic combination that brings up some very interesting thoughts actually on, like, think about this for example. Extreme black is supposed to dilute any yellow fault hairs to white. So what would recessive yellow do to it? Or what would extreme black do to recessive yellow? If they can both be homozygous at the same time, perhaps recessive yellow would be diluted to white. You know, it's a question. We're not sure though, since that's not been tested very well. But then, let's see. As far as chocolate goes, it's hard to say since it doesn't dilute yellow very much, and most of your mice look to be yellow. So we're going to put some stars here, because we're not sure what's going on with that locus. Some of your mice look like they could be blue diluted, so we could put B question mark, because what blue dilution does to yellow pigment is it actually makes it paler and more creamy. It's what we call straw in the fancy. So I can't really color it once again, but I'll draw a little cute mouse. I think one of your long hairs look kind of pale and creamy, although I'm thinking that C dilutes are probably responsible. But it'll create like a straw sort of appearance. If you look up like blue brindles, you'll see some pictures and look at the yellow pigment, you'll see the straw-like color. For the C locus, I'm pretty sure you've got some C loci stuff going on. So chinchilla, or the chinchilla dilution, has a pretty strong effect on yellow. It can either be complete or it can be mild, and it will create anything from a cream to a stark white depending on what modifiers are present. Modifiers are just genes that slightly adjust color or pigment in darker. Like, you can have darker or lighter versions of a black self. Not all black selves look the same. So that's something to keep in mind. And it's the same thing here. Not all chinchilla diluted recessive yellows are as yellow or as white as each other. And then extreme dilution has a very intense effect on yellow. It will dilute it completely to white. So if your mice were extreme dilution or completely extreme dilution, they'd be white mice, which could explain some black-eyed whites if you know that extreme dilution is possible. A yellow mouse that's got this combination will usually end up being a white mouse or a bone mouse or some other combination. And then Himalayan, this homozygous is Siamese, and it has a decently strong effect on yellow. It does turn it into white in my experience. So also we'll probably get you like foxes, snows, stuff like that. And of course albinism, just lowercase c, it's going to make everything completely white no matter what color it is. And then pink eye dilution has a subtle effect on every color. It turns black into silver. It turns blue into an even paler silver, sort of, or I guess black into dove, blue into silver, chocolate into like this kind of creamy in between color, yellow into like this really pale soft color. So very interesting. Oh, and I wanted to let you know, one of your mice is satin. You could probably tell, and that's what, like the first mouse, I think the female you showed, is what I'd call a recessive yellow Angora satin. So they've got like that Angora coat, and they've got the pied markings, and the recessive yellow, you can clearly see that it's shiny, so, and it looks just to be a standard recessive yellow without any dilute, so that's what I think you've got there. And I'll probably just comment on the rest of your mice to show you. But I think that's the most important thing. If you want to guess at these, you can come up with Punnett squares for all of the possibilities and write down what they'd look like if they had this combination of genetics. And that will help you plan out what pairings you can do to test breed. And what you'll want to do is find another mouse that may or may not bring out whatever you want to figure out. So if you want to figure out what C dilutes might be at play, what I'd recommend is finding a known C dilute mouse and pairing them and seeing what you get. So if you pair like a Siamese or an extreme dilution, and you know that they're an extreme dilution to one of your mice, and then you get babies that look like the results of an extreme dilute, then you'll probably know that you got extreme dilution. It can get a bit more complicated than that, but hopefully that is helpful. Sorry for the length, but there you go.